Next, we will start to discuss the different factors that affect genetic diversity. We'll start with genetic drift. And as we go through these next couple videos, I've put them into discrete videos for the sake of time, but keep in mind that all of these factors are happening at the same time and they can be influencing each other. So it's not like just drift is happening and then in the next video just natural selection is happening. Remember that none of them are acting in isolation. They're all interacting with each other. So what is genetic drift? Genetic drift is a change in allele frequencies over time, so across generations, for instance, specifically due to chance. So often the example that's used to illustrate this is thinking about a quote-unquote population of M&Ms. Each color is a different allele. If you were to stick your hand into that bag of M&Ms, pull out a handful to eat, if it just so happened that you only got blue ones, that's not due to any type of adaptation, or it's not because the blue ones were better fit to survive in the environment. It's just completely due to chance. You happen to pull the blue ones. So when we're thinking about genetic drift, remember it's due to chance, and what the type of change that it results in is non-adaptive. So in your book, it talks about how it's basically individuals that die and don't pass on their genes are the ones that are in the wrong place at the wrong time. They use an example of flowering plants that are killed by trampling animals, which made me think of The Lion King. I think at least a few of you have mentioned that you like Disney movies. So if we ignore the whole scar manipulation factor here, if we think about Mufasa being trampled by the wildebeest, which still is like one of the saddest things to ever happen in a Disney movie, he didn't die because he wasn't adapted to his environment. He just was in the wrong place at the wrong time, and that's why he didn't pass on his genes. So genetic drift is changes in allele frequencies that are happening just due to random chance, and what it results in is non-adaptive change. And the overall, first of all, the result and which populations drift have a stronger effect in, the overall result is that genetic drift decreases genetic diversity, and it does this more so in small populations. So if we look at this example here, on the left we have a large pool of individuals, and on the right we have a small pool of individuals. And if we look at this small pool of individuals on the right, what's happening over time is that we are losing these 3 individuals. If we lost 3 individuals in the large population, it wouldn't really matter because we have other individuals that are still carrying that same lowercase a allele. But what's happening in the small population, if we lose those 3 individuals, what we've done is essentially wipe out that little a allele. We don't have any individuals left that still have that allele. So if we looked at our new population now, what we have is called fixation, where the only allele present is that capital A. And so that's why genetic drift overall has the effect of decreasing genetic diversity. We're actually losing alleles, and it has a much stronger effect in small populations. This process, or this idea of genetic drift, is the basis for calculating different things, metrics that are useful in molecular ecology, one of them being effective population size, which is often abbreviated as N sub E. And we'll talk in the next slide about what effective population size is, but one thing I want to emphasize that it's not is the number of individuals in a population. So effective population size does not just equal the number of individuals. So in these images here that I took from the San Simeon elephant seal colony in Central California, the picture on the right, it blows my mind. These adult males are 16 feet, 5,000 pounds. They're huge. But the idea is that if you were to look at this colony and count all the individuals that are present there, that is not the effective population size. So just to be clear, that is different than what an effective population size is. But what is effective population size? For me, I have a hard time thinking about this and explaining it, and the easiest way for me is to use an example like I have written here. So if we have 500 individuals that are losing genetic variation at a rate that would be found in an ideal population of 100 individuals, the effective population size is 100. So again, remember that effective population size is not just the number of individuals, so it's not 500. And so, okay, what do we mean by ideal population here? An ideal population would be a population where we have random mating or equal numbers of offspring per parent, for instance. So if we go back to that elephant seal example, based on what we know about elephant seals, we can say that elephant seals have a lot of these characteristics based on their reproductive strategy that would mean they're not ideal, right? So we have these males that fight for access to different females, so some males don't get to reproduce at all. The mating definitely isn't random because the females really only are allowed to mate with the males that have established their territories. So there are differences between an ideal population and how many individuals we have in our calculated effective population size. And again, we said effective population size is based on this idea of genetic drift. So when we're thinking about losing genetic variation, remember this is based on the fact that a smaller population size is going to be more prone to drift and see larger changes in allele frequencies through time, which often results in a loss of genetic variation. So when we're thinking about how to actually calculate effective population size, you can calculate it from molecular genetic data using something called the temporal method. And so the idea here is that you collect from two different populations throughout time. So say time point 1 and time point 2. And those need to be separated by at least one generation. And then what you're doing to compare those two different time points is seeing if allele frequencies changed. And remember that genetic drift causes allele frequency changes more rapidly in populations with small effective population size. And so the idea here is that if we see big change in allele frequency, that means that's likely a small population with a small effective population size. If we see, oh, and I should note that triangle that I'm drawing is how I abbreviate change. I think that's actually a convention. I didn't make that up. I got it from somewhere. And then if we have a small or no change in allele frequency, that means that drift isn't having a strong effect, and so we likely have a large population with a larger effective population size. So that's the overall way that we interpret this type of data. So let's walk through one example of this. They've actually done this technique in yellow-eyed penguins. So what they did is for their contemporary samples, they took blood samples from contemporary live penguins. So I'm not going to try to draw a needle, but they drew blood from penguins. For historical samples, what they did is they actually looked at toe pads from museum specimens. So those are their historical samples. That's often the way that people are able to get samples across time, is through using preserved museum specimens. And they used 12 microsatellites specifically to look at allele frequencies and whether they changed between the historical samples and the contemporary samples. And what they found was really large changes in allele frequencies. So does that suggest a large or a small population size based on what we just talked about? So if you said small, you are right. So if you have a small population, drift will have a larger effect, which leads to bigger changes in allele frequencies between your historical and contemporary samples. So this was actually a cause for concern. This is a species found in New Zealand, and given that it has a low effective population size, the use this information to help inform conservation efforts in terms of monitoring, to think about how they might deal with climate change, disease outbreaks. So very similar to what we talked about with the koalas. Speaking of our friend the koala, another benefit of using this temporal data approach is that it can inform conservation management. So what I mean by that is this is a screenshot of the results and conclusions summary from this paper I've cited here. What they did in this paper with the koalas is use historical museum samples. So again, that's often the way you can look at allele frequencies from the past. They specifically used mitochondrial DNA haplotypes. And again, they used something else we talked about. They used rarefaction to account for differences in sample size. And overall, what they found is that the historical samples actually had low mitochondrial diversity. So they concluded that even before population decline started happening, these koalas already had low mitochondrial diversity. And this is informative because we still should be worried about koalas, but this is maybe one less thing to worry about. So if throughout time we saw that their mitochondrial haplotype diversity decreased, that would mean they're probably less likely to adapt to changes right now. But if they've had this low mitochondrial diversity for a while, even before humans started really affecting them, it might mean that they possibly will be able to continue to persist because they have been doing it throughout time, even with this low mitochondrial diversity. So in general, if we compare historical and contemporary samples and find that the historical ones also had low genetic diversity, we're less worried about that particular population. Again, we should still be worried. It's one less thing to worry about, which I'll take at this point. In contrast, if we compare historical and contemporary samples and find there are large changes in diversity and effective population size through time, that's when we're even more worried. And that's because if we used to have really big population sizes, really big diversity, large diversity, and now we don't, again, that's another indication that perhaps something has happened to this population where now it is really not well suited to be able to adapt to future conditions. This is actually something they found in the Seychelles warbler. I chose these pictures in particular. I spend way more time than I should picking individual pictures for my lectures. I chose the one on the left because the bird's doing the over the shoulder, like, what are you looking at thing. The one on the right is doing the sassy, make sure you get my good side thing. So that's why I chose these two pictures of the warblers. But in this paper cited here, they found that throughout time, there were really big changes in diversity and effective population size, which is concerning because this indicates there was a rapid loss of genetic diversity, and the surviving genotypes might not be well suited to be able to have the individuals survive future challenges and changes in the environment.